Yeah, the subject is quite broad, because there are multiple ways that microbiome data can be analyzed, and moreover, they can be produced. So in this discussion, I have tried to, let's say, put everything into a framework that other people can also follow. Um, and I put more weight on some tips, or some very sensitive issues that have to do with the analysis of the data and the statistical analysis of the data. And these points are quite important because they can really change the results and possibly the associations with disease. So the microbiome, as everyone knows, is composed of bacteria, viruses, prokaryotic and eukaryotic viruses, microbial eukaryotes, and archaea. Generally, our ability to detect them is based on whether these microbes have or share a ribosomal gene. So for species who have a ribosomal gene, we know that 16S-based sequencing is being used to detect them. But in this way, of course, we miss the viruses which do not share, let's say, genetically homologous regions. So based on this, metagenomics is the way forward, because it's a technology that allows us to detect both microbial eukaryotes and bacteria, archaea, but also viruses. And this is because it's an undirected approach, so it's unbiased. We don't use, at least in our setting in Cure, we don't do a pre-amplification with a certain set of primers. So our aim is to analyze directly the genomes that are present in the tissue that we're interested in, and in Cure, of course, this is the nasopharynx. So generally talking, metagenomics is the direct isolation and analysis of genomic information contained in an environmental sample. So this can be in the environment, but also can be a certain tissue in the human body. And exactly because we isolate nucleic acids, we can isolate DNA and RNA. From the DNA, we get the information about the taxonomic capacity of the assembly. So which species are present there. And of course, we can get information about the genetic composition of those species. And with RNA, we can also get information about the taxonomic capacity. This refers to the RNA species, for example, RNA viruses. But importantly, we can get information about the taxonomic and transcriptional activity. So basically, the genes that are being encoded in the environmental side. And this is called also metatranscriptomics. So the term microbiome basically is a technical term, and the best way to specify it is a collection of microbial and viral taxonomic lineages within a metagenome, because essentially this is what we're identifying. We're identifying taxonomic lineages. So the information we're getting out of it, we are not sure, if it corresponds to a certain microbe that is currently present in the sample, or even if it has a replication capacity. So based on this, on the information we can draw from metagenomes, we can study different characteristics of the microbiome. So this include, for example, taxonomic diversity. So in ecological terms, how diverse is the sample, or how similar are two different samples or groups of samples. We can characterize the evenness and the richness, how many different species are there, and of course the composition. We can get information about the functionality, including the signaling ways and the metabolic pathways. We can study how genetic information is transferring from one species to another, either through horizontal gene transfer or through mobile elements and plasmids. We can characterize ecological interactions, for example, the generalist versus specialist species, like the ones we study with Nina regarding the bacteriophages, or in general, ecological interactions between microbes. For example, they can be commensals, or they can be competitors. We can also describe the genomic content of each species, but also the community. Basically the dinucleotide odds ratios of all the genomes we identify. And all of this, we can use all this information to try to describe the structure of the community. And sometimes microbial networks and systems biology is being used in order to achieve this. But more or less, all of these characteristics, we need to keep in mind that they are the main way how the microbe interacts with the human host. And this is a two-way interaction. So starting from I will just say a few words about the laboratory aspect, so the wet lab procedures. There are many different ways that we can identify metagenomes, and this depends on whether we are interested into describing the bulk DNA content of the environmental site, or if we are interested into certain species or certain types of genomes, for example, viral genomes or viral metagenomes. So in our case, in Cure, we do shotgun sequencing of microbial metagenomes, which means that we isolate DNA and RNA, and then we do a direct high-throughput sequencing. The output we get is next generation sequencing reads, which we use in order to classify these reads and identify which species are present in our metagenomes, and try to characterize as much of their genomes as possible. So in this slide, we have a typical flow diagram of a metagenomic project. Actually, these are only very basic steps. The actual pipeline is quite bigger than this one. So everything starts with experimental design. Then we go in the dry lab procedures, which include the sampling, the fractionation of the sample, DNA extraction and RNA extraction, and of course, sequencing. The wet lab is based mostly on the cleaning of the data and then performing assembly of the contigs or direct sequencing read classification, annotating the sequencing reads and the binding. And in the meta-analysis, essentially, is the whole package of doing comparisons between groups of interest and modeling of the microbial communities. Something really important for these pipelines is that they constantly change, and this is because each project has its own, let's say, characteristics, and we have found out that usually it's not very usual to take a certain pipeline and use it exactly the same in another project. So they're, let's say, customized pipelines. Of course, the tools that we and other people use, they can be found, most of them, on the internet. So the experimental design is very important in microbiome studies. So starting point should be a small-scale experiment in order to understand the magnitude of the variation of the system, and this includes the technical and the biological variation. For example, small number of samples could be used and sequenced to a shallow depth, and then analyzed to determine if a larger sampling size or greater sequencing effort is required to obtain statistically meaningful results. And this is very important. We'll see also in the following slides we'll discuss about the sequencing depth. Also important is that the fact that most human microbial systems are highly dynamic. So we need to evaluate the temporal aspects of sampling, which can have a substantial impact on the analysis of the data, but also the interpretation of the data. And this is very important when we are looking at cross-sectional data, where we only have one sample per subject. And finally, good experimental design of such projects should integrate the existing ecological theories into the hypothesis that they're trying to test. So it's really important to understand the pathophysiology of the disease, if we're talking about the disease model, and try to envision, let's say, or guess how this might go along with already known ecological theories of how how microbes are assembled into communities. Now, regarding metagenomics and the sequencing technology, we always need to keep in mind that we have a sequencing detection limit, and things that we do not detect doesn't necessarily mean that they're not there, first of all. And then when we're trying to correlate sequencing reads, so the quantitative aspect with the presence of an organism, we always need to think about the different kinetics, the replication kinetics that different species might use. For example, in the first case, we might have a microbe that is being cleared from the system, and it's quite possible that at least in the beginning of this process, we will be able to pick up some quantitative data out of it. But also a species might be in the beginning of its replication activity, so it will start and picking up in copies. So whenever we're identifying certain species, we don't know, at least at the cross-sectional setting, we don't know if it belongs in the one scenario or the second one. And of course, we might have oscillations in the replication activity. For example, this is something that has been demonstrated for bacteriophages. And looking at all this, we can understand that microbiome data can contain information of past, but also current exposure. And this is equally important to integrate it in our general hypothesis about the disease that we're trying to investigate. Now, coming back on the flow diagram, apart from the experimental design aspects that we discussed up to now, we always need to think and let's say change or adapt the pipeline based on whether the study is exploratory, is data driven, or we want to test a certain hypothesis. Of course, things will change or will be adapted depending on if we have a cross-sectional or a longitudinal study. It's always good to have matched cases for all the groups that we want to investigate. And of course, this is also related to the analytical power. So we try to the power of the analysis to be higher than the technical variability. So these are really important aspects of considering even before we start the actual experiments. So sampling is the next step. Of course, we need to have standard operating procedures that are in place, they have been tested, and they are followed by all the participants. Since we're discussing about human tissue, we need to make sure that we're sampling a specific microbial niche, and if not, measures need to be taken so we make sure that we don't get contamination from other microbial niches. And we will see in the following slides the effect of the biomass in relation to the yield of the microbial DNA that we can pull out from the sample. For sample processing, there are ways where we can enrich for the microbial component of the biome. And this is mostly used in tissues where the contamination with human sequences is quite high, for example, like the one we get in the respiratory tract. And of course, we need to evaluate the different enrichment methods and evaluate their effect on the composition of the microbiome, and of course, normalize all the different samples to the same level and make sure that we don't have any batch effects. The nucleic acid isolation is quite important. There are many different ways, different kits that they are available and someone can use, or even mechanical manual processes. It has been shown that a major source of contamination in a microbiome project is the step of nucleic acid isolation, and that's because the buffers are not sterile, so it has been shown that they contain a specific signature. And of course, over here, we want the best possible yield and the best possible quality of nucleic acids. And finally, an optional step of nucleic acid amplification if the yield is quite low, and this is usually because of the initial biomass. So when the initial biomass of the sample is low, then the nucleic acid yield is also quite low. In some cases, we need to do a nucleic acid amplification. In Cure, we do a whole genome amplification by using multiple displacement amplification. And of course, as we have done in Cure, we need to test the effect of these amplification processes on the composition. So for the nucleic acid amplification, there are many different approaches, so molecular methods that someone can use. The ones that have been broadly used in metagenomic projects are the ones in the squares. So as I've discussed, we use the Find29 DNA polymerase based amplification. So there's a big toolkit that someone can pick from. Now we'll touch base, let's say, one of the big problems of microbiome data analysis. And this is the fact that the microbiome data are compositional. And believe it or not, this creates many issues in the statistical analysis also of the data. So why microbiome data are compositional? That's because we use specific platforms in order to sequence our metagenomes. And of course, each sequencing platform has a certain maximum output. So we have a ceiling of the possible number of sequencing reads that we can use. And in this table, I have just pulled out the comparison of Illumina short read sequencers. And you can see the different outputs. So what does it mean that we have a maximum output? It means that imagine that the microbial community or the microbial niche is, let's say, a lake, and we have a pint glass which can have a certain capacity, and we just dip the pint glass in the water, and we get a certain fraction out of the water, and we just hope that the composition of the water in microbes within the pint glass is equal to the composition of is similar to the actual composition of the microbial community in the lake. So that's the major problem in microbiome data. And another aspect of this issue has to do of the ratio of human sequences versus microbial sequences that we pull out from the human tissue, from the environmental side. And as we can see in the graph on the left, different tissues, they show different contamination of human sequences. For example, the gut microbiome studies that are based on stool samples, we can see that we hardly need to do any enrichments for microbes because the human contamination is really, really low. So most of the sequencing output that we get is directed towards non-human genomes. When we compare these, for example, to saliva or the nasal cavity, and as we have seen the nasopharynx in Cure, things are quite different. Over there, we have about 90 to 99% contamination of human sequences, which means that if we use a sequencing technology with an output of 1 gigabyte, only a very small percentage of these sequences will carry microbial information. That's why it's really important, and we have spent actually a lot of time in Cure in order to try to drop this ratio, so drop the contamination or lower the contamination of human sequences. And right now, I think we're working around 70%. And with this 70%, we have actually achieved to get quite a bit of microbial information, which covers all the different four components of the microbiome, so bacteria, fungi, or microbial eukaryotes, viruses, and archaea. In Cure, on average, we get about 15 gigabase pairs of sequence information per sample. And something that is really important to understand is that this doesn't mean anything if we don't analyze the data in such a way that we can understand what portion of the or a fraction of the actual diversity we are able to capture, and we can see that, for example, in the rare function curves in this graph, where on the x-axis we have the sequencing depth, so the number of sequencing reads that we obtain for different samples, and then on the y-axis we have the observed OTUs, the observed species that we capture per sample. And we can see that the lines tend to plateau, but they haven't plateaued yet. And we'll see also in the following slides that this is something that is quite common in metagenomic studies, and it's called the rare biosphere effect. So going back on the flow diagram of the typical metagenome project and going on the, let's say, the dry lab aspect, the user need to pick between different sequencing technologies, also different platforms. To date, for metagenomic studies, the most widely used are the 454 from Roche, Illumina, the solid sequencer, PacBio, and Nanopore is also trying to get in the game. So the major things that we care about is the output, so the number of sequencing reads, the length of the sequencing reads, because these are directly related to our ability to assemble larger contigs, and of course the ratio of error, the error ratio of the technologies. For the assembly, which I will not go through all the different options we have, but the assembly process refers to the recovering the genome essentially of uncultured organisms, or obtain full-length CDs for subsequent characterization. Different ways. There are different ways that we can do that we can assemble genomes. They can either be reference-based assembly or co-assembly, or they can be de novo assembled genomes. So two tips here that the user needs to take into account is that most, if not all, microbial communities include significant variation at a strain and species level, which makes the use of assembly algorithms that assume clonal genome less suitable for metagenomics. And that's because in this case, the assembly algorithms might not be able to discriminate between very similar genomes, and they will assemble them into high-order taxonomy level. So if you have two strains that are quite similar to each other, most probably, at least with the traditional assembly algorithms, they will be referred to as one species. So we lose basically a fraction of the heterogeneity of the metagenome. But now there are new assembly algorithms that do not take into account the clonal genomes, so they are being used in a much better way in metagenomics. And the second tip is that rare k-mers do not represent sequence errors as initially assumed, but instead represent reads from less abundant pangenomes in the metagenomic mix. So this is particularly important when we are evaluating the composition of the metagenomes, and we try to rank the metagenomes based on the abundance of the species. So up to date, most clustering approaches or most exploratory approaches that aim to identify differences between groups of metagenomes or groups of microbiomes, they are heavily biased towards the dominating species in the community, which on its own it raises a lot of questions regarding the effect and the actual role that all the different species play in the assembly of the microbial community. But nevertheless, we need to keep in mind that whatever we observe in low frequency and in low abundance is not necessarily an error of the sequencing process. After the assembly process, we have binding. So binding is the process of sorting DNA sequences into groups that represent an individual genome or genomes from closely related organisms. And basically, we have different ways to do the binding. Compositional binding makes use of the fact that genomes have covered nucleotide composition. And this also be reflected in sequence fragments of the genomes. And the unknown DNA fragments might encode for a gene, and the similarity of the gene with already known genes in a reference database can be used to classify and hence bind the sequence. So this is pretty straightforward. And finally, the annotation. Essentially, metagenomic annotation relies on classifying sequences to known functions or taxonomic units based on homology searches against available annotated data. So from all these, apart from the de novo assembly process, we understand how important the reference database is in our ability to identify sequences, but also to make sure that the sequences are not biased towards specific components. Unfortunately, the reference databases up to date, they are quite heavily biased towards certain microbial, bacterial genomes, and this has to do with the research that is being performed all these years. So there are actually dozens of available software to use for metagenomic or 16S-based sequencing approaches, and also for viral metagenomics. And these have been really nicely reviewed in this paper by David Robertson and Avram Tapinos, which are members, of course, of CURE. And one of these tools is the one we use as part of our computational pipeline. It's called Kaizu. And essentially what we did is we have adapted Kaizu, and we have integrated in a larger pipeline, which starts with the processing of the raw reads, so basically cleaning up the reads, evaluating their quality, removing human reads, because of course we're interested on the microbial component. And then we use Kaizu to do a first reference-based classification of our sequencing reads. Essentially, we cluster our sequencing reads into groups that correspond to the microbiome components, and then we do co-assembly of these reads for each one of these groups. So there are many different options. There are well-established also assemblers and software tools that can be used, and they have been reviewed quite extensively in the literature. So I will not go into any more details on that. So a very important aspect of the, let's say, metadata processing is the normalization of sequencing read data, and you will see that this can actually have a detrimental effect on the associations or the statistical analysis we're doing. So in CURE, I think we use quite a strict way of normalizing our data. So we do multiple normalizations. First, we do at the sequencing depth. We also do genome size normalization. We normalize at certain taxonomic levels. And on top of that, we also use some thresholds of our own, which have to do with either the contact length, the number of reads, or the incidence or the frequency of observation, etc. But more importantly, in CURE, we evaluate all our data, all our results using different scenarios of normalization processes. So we make sure that the data are quite robust. And whenever we do normalization, we always need to know that there's always a trade-off. And exactly this is why we should also evaluate results in different scenarios. Now, we'll say a little bit more about this trade-off. So the sequencing depth. So as we discussed before, the number of metagenomic sequencing reads usually varies between samples. And of course, this can affect the downstream analysis, for example, the alpha or the beta diversity estimates, or the relative abundance, or differential relative abundance. And this because, as we see, as sequencing depth increases, more species are discovered. So the mostly used normalization procedure is called rarefying, where basically we subsample from our sequencing reads, and we use an equal number of reads, which is subsampled from each sample. So basically, it's setting up a certain threshold to all samples where we can analyze the data. The trade-off is that the rare function is by doing rarefaction, we get rid of many different species that are observed in a low number of sequencing reads. So this is not an ideal scenario, but nevertheless, this is something that it has been widely used. And of course, the trade-off is inclusion of more samples or retaining a higher sequencing depth. That's the actual question that we need to answer. Let's have a look now on the effect of different normalization processes in the composition of the microbial communities. So in the first graph over here, we see the raw, let's say, composition of our microbiomes in the 4 microbiome components: viruses, bacteria, archaea, and eukaryota, for different samples. And this is the raw sequencing read. So basically, we have annotated the sequences, we know which species are there, and we just plot them to see the composition on the 4 microbial components. On the second graph, what we have done, we have performed a genome normalization. So under the assumption that species that contain larger genomes are more probable to be detected by our sequencing technology, we have, and actually this was in help with of Joe Busby in Glasgow. So we do a genome normalization of our data, and as you can see, after doing the normalization, we have a higher composition in viruses, and that's expected because viruses they have much less. They have much smaller genomes than the bacteria and the microbial eukaryotes. And on top of that, if we also do a rarefaction, we can see how the composition will also change by losing some of the viral species in the community. So this is something that we always need to keep in mind, that whatever kind of normalization we do in the sequencing data, we always jeopardize of losing certain species. And that is why we need to evaluate different normalization processes in relation to the robustness of our statistical tests and disease associations. And in literature, it has also been pinpointed another problem with specifically with rarefying data, is that both proportions, so the compositionality, as we discussed, of microbiome data, but also the rarefied counts result in a high rate of false positives in tests for species that are differentially abundant across different samples. And we can see this in this table on the right, where in one case, in the first table, we have the raw sequencing data. So we have the count data without performing any kind of rarefaction. And on the table on the right, we have the rarefied abundance. So everything has been rarefied to 100 sequences. And if you do a t-test, basically, between the first case and the first scenario and the second scenario, we see that one scenario gives us a statistically significant change, whereas the other one doesn't. So as you can see here, there's a big problem with the first steps of normalizing the sequencing data to the same level, so we can compare essentially samples belonging to different subjects. Another problem also has to do with the clustering. So the rarefying procedure, it often discards samples, as we discussed, that can be accurately clustered by alternative methods. So apart from rarefying, we need to look for other alternative methods in order to do normalization of the sequencing of the data. And this is something we also do in CURE. So in CURE, basically, we have evaluated, I think, 4 different normalization processes, and we run the same test again and again in the different using different normalization thresholds. So how does the microbiome data actually look? So the output metadata, they basically can be reduced to tables. And this is an example of such a table. So the columns represent samples, and the rows indicate either taxonomic group or a gene function in terms, in cases where we do metatranscriptomics. And the fields contain the abundance or the presence absence data, or the raw number of sequencing reads. And as we discussed, the rows carry, at least in this case, we have the taxonomic lineages. So this is the first level, let's say, of metadata that we obtain from after our computational pipeline. And what do we mean by taxonomic lineages? So basically, for the majority of environments, many organisms will not have been taxonomically classified and sequenced before, or the resolution of the sequencing reads that we get from our experiment is not that high. So in this case, in both cases, the list of taxa may have to be generated at a low resolution phylogenetic level, for example, at the phylum level. In order to achieve, of course, a reasonable proportion of classified reads. And this is an example of that. So, for example, I just pulled this from our Cure data. And it's we have different taxonomic lineages that have been identified in different samples. And if we look at the last lineage, this is where we have the highest resolution because we're able to classify that sequencing reads up to the strain level. So we know that sequencing read belongs to that strain. But as you can see, we have other sequencing reads that have been classified up to the species level, or up to the genus level, or even up to the family level, and so on. Moving up to the group level, which is even above the phylum level. And this creates some inconsistencies in our final data set because we have sequencing information essentially that carries different resolution thresholds. And of course, someone can take the whole data set and analyze it as it is. But in that case, we don't know whether, for example, the read that has been classified as Bacteroidetes uniformis actually belong to another strain than the Bacteroides uniformis strain that we see in this table. So that's the real problem that we have to address. And how do we address this? Basically, we normalize the lineages to the same taxonomic level, and this can be, of course, can be decided a priori, or you can build a computational pipeline where you get automatically this type of normalization. This is what we have in Cure, basically. So the data we start analyzing at the species level, which means that all the sequencing reads that have been classified to below the species level, they are summed up to the species level. But also we use higher taxonomic levels. So basically we go usually up to the family level. And of course, it's equally important to say that we need to analyze different taxonomic levels whenever we detect a difference between groups or between the samples of interest. And that's because we need to evaluate whether the difference, at least in the relative abundance, but also in beta diversity, they are due because they're being observed due to a certain species, or there is a generalized effect, that it is being related to a whole family of species. So the next step is to, so from here and until the end, I will basically discuss microbiome features that are typically investigated in microbiome and metagenomic studies. Of course, this is not the full extent, of the things that someone can look for. And we can understand that because in Cure we have, 3 different work packages that are, are looking at different things, different aspects of microbiome. But let's say this is the standard, starting point. So we have the ecological indexes. These include the richness, so the number of species that we observe in each sample. Then we have diversity indexes such as Shannon, Simpson, Chow index, and others, and they describe the diversity within a sample. The difference between richness basically and diversity is that richness is the actual count of the number of species that we get, whereas the diversity takes into account also the relative abundances of each species, but also the richness. Then we have beta diversity. So beta diversity essentially is the comparison of samples based on their composition. And over here, basically, we use distant approaches and ordination methods to plot them into 2D or 3D plots. And the most widely used are the break areas, Unifrac, which also takes into account the phylogenetic distances between the species. However, Unifrac is really difficult to implement. It's quite difficult to implement in metagenomic studies where we also have viruses. And the Atkinson distances, which we also use in Curie. So basically in Curie, we use the break areas and the Atkinson distances. Then, of course, we characterize the abundance and the incidence of each species. So abundance is, as we discussed, the relative number of sequencing reads within the metagenome, and the incidence is the frequency of detection, so how often we observe the same species in our cohort or in groups of samples. Then microbiome studies essentially are almost synonymous with clustering and modeling approaches. And we'll discuss about this also in the next few slides. What we need to keep in mind is that metagenomic data contain many more species or gene functions than the number of samples taken. So they are high-dimensional data, and all the different approaches that are referencing, using and analyzing high-dimensional data are certainly applicable to microbiome studies. Of course, take into account that we have this compositionality aspect. Now, there are package tools or software tools that provide, let's say, information about most of these factors, such as the ANOSIM, Microbiome, Metastats, primary package. So these are highly referenced, and they have been used in many different studies, and they can provide a very good level of exploratory analysis of the microbiome data, especially as a starting point. So for someone who's setting up, let's say, a protocol for the first time, these are really, really good. So I said something before about the rare biosphere effect. So this is so I will describe now why this is actually something that we need to take into account and use appropriate approaches. So what is the rare biosphere effect? Is the fact that in metagenomic studies, and regardless of the human side or the environmental tissue, the environmental side, the observed diversity is quite high, and most species are observed with low abundance. This is something that it can be described as a power law distribution in our data. So if we graph the identified species on the x-axis and the relative abundance of those species on the y-axis, what we see is that we have a very narrow subset of species that are observed with high relative abundance, and we have many different species that are observed with low relative abundance, and this also stands for the frequency of this species, the frequency of detection of this species. So there are two consequences of that. One of them is the one described in this slide, and it is that even with very large read numbers, we will have only sampled a fraction of the true diversity. So this is something that we need to take into account in our microbiome study. So the things that we describe, basically the things that we detect, is not the whole truth. There are many things out there. And as a result of this, in Cure, for example, we get a huge number of different species. So basically classified at species level or below, coming from all the different microbiome components. So from phase one, from the phase one cohort up to now, I think we have identified more than 10,000 species. So that's a huge number, and it's basically because of the rare biosphere effect. Now there are many in microbiome studies. We use a lot of exploratory statistics in order to search for natural patterns in the data, and multivariate ordination techniques are, let's say, number one or the method of choice. So basically, samples are positioned in a space of reduced dimensionality, so as to preserve the distances between them in the original higher dimensional space. And this is usually achieved by doing principal component analysis of Euclidean distances, or principal coordinate analysis of unifrac or Brachiotis distances. So over here, I just have an example of the Euclidean distances. So on the matrix on the left, the heat map, this is basically similarity matrix. So we compare different, we do pairwise comparisons of the different samples between them, based on their microbial composition, and then we use the similarity matrix in order to annotate the distances between all the different samples in principal components. Or you can do that in a two-dimensional plot. We only have two components. It's the same thing. So a big part of the analysis of the metadata is to evaluate these distant metrics, and actually, because there are different metrics, different metrics can produce different results. It's always good to evaluate multiple distance metrics. So in Cure we use the as I said, the Brachiotis and the Euclidean distance. So they're both and the Atkinson distance. So Atkinson distance is a Euclidean distance, whereas the Brachiotis is not Euclidean. So we evaluate both of them in order to identify differences between groups of samples, or between health and asthma in our case. And clustering is also equally important in exploratory data analysis. So essentially with clustering, what we're trying to do is search for natural groups or partitions in our samples. The most widely used, of course, is hierarchical clustering, where a tree of relationships generated without explicitly grouping samples, unless an arbitrary cutoff is chosen. And this can be seen, for example, this cutoff in the on the cladogram on the right over here. So it's up to the user, where to put this threshold, and depending on where someone assigns the threshold, it will change the number of clusters that we'll get. So these types of hierarchical clustering is very we see it quite often in comparisons of the relative abundances between microbiomes. But they're not necessarily the most appropriate way to cluster the microbiomes. Now, another question, or a target, or a target that most microbiome studies the past 5 years aim for, including Cure, is the question of the natural number of types of communities that are actually present in the microbiomes. And this actually received particular attention in the context of the human gut microbiomes and the enterotypes. So basically, the enterotypes of the human gut microbiome are essentially clusters of microbiomes that have been named as meta-communities or microbial communities. So they are basically a project, a process, a byproduct of, they are a product of clustering approaches. And finally, we have the classification or unsupervised learning, which is closely related to clustering, but it's not exactly clustering. So the problem we address by doing classification is to find natural groups in the data. Is to predict the group of a new sample given a labeling of samples in a training data set. So we have a training data set, and based on that, we try to assign a new sample into one group. So these are three ways, groups of analytical tools that someone can use in microbiome studies. And actually, we use all three of them in Cure. So, but the sensitive aspect is to pick the appropriate approaches in order to achieve your goals and to address these questions. And unfortunately, there are inherent problems in using standard multivariate techniques for the analysis of metagenomic data. And this is because there are three main reasons for that. First, the data, even if normalized into relative abundances, are fundamentally discrete, so they can only be approximately modeled by continuous variables. The second one is that the high diversity results in a very sparse data set. So as we discussed, most taxa appear in only a few samples and at low abundance. And finally, the samples vary in read number. So a small sample will inherently be more noisy than a larger one. So all these issues can be addressed using an explicit sampling scheme. So fortunately for us, there are ways where we can achieve a very robust classification and clustering of our subjects based on their microbiomes. So specifically here we use the Dirichlet multinomial mixture models, which is a probabilistic modeling essentially of probabilistic clustering of our microbial of the microbiomes. And in order to understand how these, these approaches can affect the endpoint, so basically the statistics, the statistical testing, over here we see how the relative ability of competing methods to detect true positives, so true positive differential relative abundances, basically, in simulated data. And in this graph specifically, there is a comparison. So all four graphs, they represent a different methods. So A is ANCOM, B is DSIC2, C is EDGAR, and D is Wilcoxon. So I will not go into the details of what these four things do, but all these are compared to the Dirichlet multinomial mixture models. And for example, you see in each graph, you have a diagonal line. So whatever is above the line and the number is basically the true positives that DMM managed to detect in relation, to the specific approach. And what we can see here is, apart of, from the superiority of DMM, is the fact that the variability between the different approaches. So different analytical methods we give or will influence somehow, the list of the final list of species that are being observed with differential relative abundance between the groups of samples of interest. So the take-home message is basically that the statistical approaches in general should take into account the discrete nature of the metagenomic data, the sparsity, and the variable size. And so in order to, and I think that's the final slide to conclude. Everything starts from the biological question. So before setting up the metagenomic project, someone needs to carefully evaluate the pathophysiology of the disease, if we're discussing about disease, how that might reflect to the microbiome, what's the temporal variability or the heterogeneity within the disease in terms of the characteristics of the subjects, and then try to envision whether this could let's say walk along with an already established ecological theory that could explain or could create this association between the human host and the microbiome. And based on that, we can someone moves forward, designs the experimental pipeline, and these can be an assembly basically of already published tools. Then test the whole pipeline from sampling to the statistical testing on a pilot study or in a simulated data set, specifically for the annotation algorithms and the assembly algorithms. And after this, after a first pilot study, this will feed back again on the experimental design. So most probably the user will have to, change the experimental design a couple of times in order to get the ideal performance. After that point, someone can move on a larger study, and then, of course, do the analysis. And as we discussed, need to have specific to be very careful on the normalization methods, statistical methods, and the clustering/modeling approaches. And of course, all these again should try to step, let's say, on the grounds of a very robust ecological theory. So these are, I think I've finished. I don't have anything else. Yeah. So thank you for having me and having this discussion.